All right. So in this video, we're going to talk about the anatomy of the male reproductive system. And so the primary sex organs for males are the testes. Those are the gonads, and they produce gametes, which are basically sperm. And the accessory organs include a complex of ducts and tubules that lead from the testes all the way to the penis and the external urethral orifice. Now, the penis is what we call the organ of copulation because it's necessary that it engorges with blood during sexual arousal for copulation to occur. Now, what we see here is a mid-sagittal cut through the pelvic area. And so just for reference, you guys remember we have our mons pubis over here, pubic symphysis, urinary bladder, just like in the female system. However, you see that there's no uterus here. There's obviously not a vagina either. So what we have then is our urinary bladder, our urethra, which extends out to the penis, and then our rectum just posterior to that, and that leads into the anus. Now, from this mid-sagittal view, you can see quite a bit. We can see things like the erectile tissues of the penis. We can see our scrotum with some of the supporting tissues, the testi, the epididymis, which is basically the storage site for sperm. We have our vas deferens or ductus deferens, which is a muscular tube that extends up the spermatic cord through the ilioinguinal ligament into the pelvic cavity, back behind the urinary bladder, where it meets up with another gland here called the seminal vesicle or seminal gland. The seminal vesicle produces the majority of the volume of semen. It's mostly water, different buffers for acid, as well as some nutrients like sugars, like fructose and glucose. And the seminal vesicle and vas deferens meet and converge at a structure called the ejaculatory duct. The ejaculatory duct meets up with the prostatic urethra, which is right here within the prostate. The prostate also is a gland that produces some of the volume of semen. It produces prostatic fluid. And so the prostatic urethra receives also prostatic fluid as well as seminal fluid and testicular fluid from the ejaculatory duct. Beyond that, we have our urogenital diaphragm. It's a little band of smooth muscle that prevents, urine or semen from leaving. But what we find then, you guys, is right near the urogenital diaphragm, we have another gland right here called the bulbal urethral gland or Cowper's gland. This produces a very small volume of semen, but essentially what the bulbal urethral gland creates is a kind of a clear, viscous type of solution that helps neutralize the acidity of the urethra. It produces something called pre-ejaculate, which is just that kind of clearish, viscous solution. Now, it neutralizes the acidity of the urethra because the acidity of the urethra could potentially damage the sperm, so it's secreted prior to ejaculation to help prevent damage to sperm. Now, beyond that, we have the rest of the urethra here is called the spongy or penile urethra. It's called the spongy urethra because you find this part of the urethra within a structure called corpus spongiosum, which is a type of erectile tissue here. And then nearby the penis, the dorsal side of the penis here, or anterior surface, we find here this is called corpus cavernosum, which actually is what engorges with blood to contribute to the majority of the erection itself. And then beyond that, you guys, we have our external urethral orifice. But the penis has different parts, right? We got our root, we got our shaft, and then the glands. And the glands can be covered with a prepuce or a foreskin right here. So we're going to first talk about in more detail the structure of the scrotum. So the scrotum is a skin-covered sac that provides the testes with a cooler environment than body temperature. Remember, we talked about how the sperm develop at a temperature that's slightly cooler than normal or average body temperature. And so in that sense, it makes sense why the testes would be located within a scrotum, which is more external to the pelvic cavity because it's cooler there. It can kind of cool off with the external environment. And it's approximately 3 degrees Celsius below normal body temperature. Now, in the scrotum, you actually find some different muscles like the dartos and cremaster muscles, which are part of the wall of the scrotum or spermatic cord, and they're involved with regulating the contracted state of the scrotum. Like if it's really cold within the scrotum, these muscles can contract, and it can help bring the testes closer to the pelvic cavity. That way they stay a little warmer. Otherwise, if it's very hot within the scrotum, these muscles can relax, and the scrotum can relax, bring the testes farther away from the pelvic cavity, and that way they can kind of cool down, and it's easier for sperm to mature in a cooler environment, but not too cold. Now, the scrotum is analogous to the labia majora in females, so effectively it's thin skin with hair. And there's also apicrine sweat glands there, so it's involved with body odor and pheromonal communication. Now, in this picture, what we see then is a cutaway view of the abdominal wall. We can see our pelvic cavity with some of our deeper vessels, like our iliac arteries here and veins. You can see our urinary bladder nearby too. Now this thick white structure here between the ilium and the pubic bone is called the ilioinguinal ligament. This ilioinguinal ligament is important because it actually is the passageway for structures into and out of the scrotum. If you look here at a cutaway view of the scrotum, what we find is that if you look at these structures, they actually have to go through the ilioinguinal ligament to either get into the pelvic cavity or into the scrotum. This is how blood vessels and nerves and the vas deferens communicate with the scrotum and deeper structures. It's through this inguinal ligament. Now the inguinal ligament has several key areas, you guys. We have our superficial inguinal ring. That leads into an inguinal canal and then a deep inguinal ring. Over here we can see it again, you guys. Superficial inguinal ring, inguinal canal, and then a deep inguinal ring. Now it's clinically significant because structures within the pelvic cavity and abdominal cavity can herniate through the inguinal canal down into the scrotum. So they're more common, but typically what can herniate into the scrotum is mesenteric fat. So fat around the mesenteries can start to protrude through the spermatic cord into the scrotum, and if there's a lot of it, it actually can impinge on proper blood flow, and that can lead to things like avascular necrosis or death of scrotal tissue, which always doesn't sound very good. Now, within the wall of the scrotum, what we find is a raphe, which is just sort of some fascia tissue. And then deep to that, we have our dartos muscle. So our dartos is actually smooth muscle, so it's under autonomic control. And it's the dartos as well as our cremaster that together help regulate the contracted state of the scrotum. That way, if it's too cold, what happens to our testes then, you guys, if it's very cold? Yeah, they elevate because these muscles contract and it draws the whole scrotum upward towards the pelvic cavity where it's warmer. Otherwise, if it's too hot within the scrotum, these muscles relax and the testes descend and they cool off, right? Now, deep within this, what we find is our testes, which is the male gonad, and it produces gametes, or in this example, sperm. The sperm are stored within the epididymis here. Epididymis is effectively the storage site for sperm. They're stored there until they're released. Otherwise, if sperm are stored long-term and not released, they're just reabsorbed by your immune system. Now, what's interesting too, you guys, is associated with our testes and epididymis is a structure called spermatic cord. So all of this is the spermatic cord, and that includes our cremaster muscle. Now, the spermatic cord, if you look deep within this cord of structures, what we find is some arteries, veins, autonomic nerves, as well as our vas deferens. Now that we have our testicular or gonadal artery that comes down and serves hot oxygenated blood it's also nutrient rich to the scrotal tissue. But then emerging from the testes, coming back up towards the abdominal cavity here, we have a series of veins that form a plexus called the pampiniform plexus. And the pampiniform plexus functions as a heat exchanger. Essentially what this does is actually the cooler venous blood going back towards the pelvic cavity actually cools down the hotter arterial blood as it descends toward the testes. That way it also helps keep the testes cooler than body temperature. Now we also have some nerves nearby which are involved, like testicular nerves, that are involved with controlling these muscles as well as sensations of pain and temperature. And we have our vas deferens, which is actually a muscular tube that emerges from the epididymis, and it carries testicular fluid up towards the posterior part of the urinary bladder during ejaculation. Now, what this picture shows you guys is in cadaver dissection. What they've done is they've cut away the scrotal tissue, so we can see our testes, the epididymis, our spermatic cord here, and then within that we can find our vas deferens or ductus deferens. We can see our testicular artery and vein, as well as testicular nerves, and they're all bundled within that. The spermatic cord is going to have those structures, you guys. We have our testicular artery, pampiniform plexus, cremaster muscle, and autonomic nerves. Testicular artery is actually going to be a branch of the abdominal aorta. We also call it gonadal artery. It descends through the pelvic cavity, down through the inguinal canal, through the spermatic cord, and then serves blood to the whole scrotum. The pampiniform plexus is a network of veins that surround the testicular artery. They're involved with cooling down the blood that's traveling to the scrotal tissue. The cremaster muscle and fascia are basically extensions of the internal abdominal oblique, and they're involved with controlling the contracted state of the scrotum to help regulate the temperature inside the scrotum. In fact, cremaster muscle is really similar to another muscle that you find on your neck called the platysma. And it's effectively a subcutaneous type of muscle that you only find in several key areas in humans. It's on the neck as the platysma, it's in the scrotum as the cremaster, and it's in the palms as palmaris longus. But in other animals, it's in like porcupines and other four-legged animals, it's a muscle that surrounds their entire body. I'm drawing a blank on the name of it. But they use that that's involved with like sticking out their porcupine quills. It's also involved with like twitching skin to help shoo flies away from fur and that kind of stuff. Humans only have it in several areas, not all over our body. But the cremaster is an example of that type of muscle. Paniculus carnosus, that's what it's called, if you guys want to Google it. Now, autonomic nerves are the nerves that basically travel with the plexus here, and they connect to the testes, so they're involved with regulating, the contracted state of the muscle you find within the scrotum, as well as, sensations of pain, that kind of stuff. Now, the testes are an oval organ. Again, it's the male gonad here, and that produces sperm. It also is involved with secreting and producing different hormones like androgens, like testosterone. There's different forms of testosterone, but this is the sex hormone that's made by the male gonads. Each testis is covered by a serous membrane called tunica vaginalis, which means the testes are actually lubricated, so that if they move around within the scrotum, they're lubricated within a serous membrane. And the tunica vaginalis has a parietal and visceral layer like serous membranes do. So you can see that here, you guys. Here's the tunica vaginalis. It's a serous membrane. So you have the visceral layer that surrounds the testes, the parietal layer on the outside. And the testis itself is surrounded by a fibrous tissue called tunica albuginea. Just like on the ovary, we had tunica albuginea. It's a dense fibrous tissue that surrounds the outside of the testes. Now, tunica albuginea also has extensions that dive down deep within the testes, and it separates the testes into a specific septum. Okay, so you have these septa that separate the lobules of the testes, and each of these lobules contain a system of tubules called seminiferous tubules. So you have lots of these small little lobes or lobules in the testes. They're separated by septa, which are extensions of tunica albuginea diving deep. The seminiferous tubules have a germinal epithelium here. So inside of the seminiferous tubule, we find something that looks like a stratified cuboidal type of epithelium. And within this, you actually have developing sperm cells. Now, these are dividing similar to how oocytes develop, which is meiosis. But then we have a lumen in the center here, which is basically the center of the seminiferous tubule. That's where the sperm cells will enter, and they get transported through the seminiferous tubules towards the rete testis to the epididymis here. And the epididymis is the storage site for sperm. So the epididymis has a head, a body, and a tail here. And the tail of the epididymis is actually what hooks up with the vas deferens, or ductus deferens, and that's a muscular tube that emerges from the epididymis and then goes up through spermatic cord. Because each testis creates millions of sperm like every minute, there's a lot of cells that need to be stored, and they're stored within a pretty complex and convoluted system of tubules here within the epididymis. Now, if those sperm cells aren't used and they have a particular lifespan, they're just basically reabsorbed by the body. What's interesting, you guys, is that because sperm don't start to develop later on in life for males, technically those sperm are actually considered as foreign cells by the body. So that if those sperm encountered immune cells in a male body, their own immune system would actually attack those sperm cells. They're considered foreign. This is one of the ways that sperm are actually resorbed and digested. Because they don't develop till later, they're actually considered as foreign cells, which is actually kind of interesting. But for this reason, you also find what's called a blood-testis barrier. There's actually a barrier, like there's a blood-brain barrier in the brain, right? There's a blood-testis barrier, and it prevents sperm cells and immune cells from communicating in the seminiferous tubules, because otherwise you get very strong immune reactions, and you wouldn't want that, right? So these sperm cells are actually separated from the rest of your body through this blood-testis barrier, and part of the cells that make up this blood-testis barrier are these sustentacular cells, or sustentocytes. And these sustentacular cells basically help not only support the maturing sperm, but they also form that protective barrier, the blood-testis barrier. That way sperm can't end up in your bloodstream, and they stay within the seminiferous tubule. Obviously, it doesn't sound like it's a very good thing if a little tadpole-y, like wiggly sperm ends up in your bloodstream, right? It sounds like it could cause some serious problems. And luckily, it's kept from getting in there because there's a blood-testis barrier. Now, the testes are surrounded by that tunica albuginea. It's a thick, fibrous capsule, and this projects to the interior of the testes at what we call the mediastinum, where there's lots of blood vessels, lymph vessels, and nerves. Now, the tunica albuginea separates testes into septa, and these septa have about 250 lobules. These lobules are full of seminiferous tubules, which is basically a big old tube full of germinal epithelium. So just to summarize, you guys, we'll go back to this picture. Remember, again, the whole testes is surrounded by a tunica albuginea. The albuginea dives down deep into the testes and separates it into lobules, about 250 per testes. And these lobules are full of seminiferous tubules. Yeah.
So the tunica
Vaginalis? Yeah.
It covers only the anterior and lateral part.
Right.
And it has the serous.
Yeah, the mediastinum over here does not have tunica vaginalis. That's where the blood vessels and nerves would enter.
And then the rest is covered by the tunica albuginea?
That's a good question. So the question was, where does tunica vaginalis cover? Well, you can see that it covers the sort of exterior portions of the testes here, and including this part of the epididymis, which you think about as being the lateral surface. Now, the entire testes is surrounded by a tunica albuginea. The entire thing is surrounded by this whitish tissue, tunica albuginea. But there's areas where tunica albuginea dives down deep, and it separates the testes into lobules, about 250 per testes. And then each lobule is full of a seminiferous tubule, which is basically a big tube lined with a germinal epithelium that gives rise to sperm cells. And all of these seminiferous tubules then meet up at the rete testis, and then that actually drains into the epididymis, where the epididymis actually is more tubules that store sperm. Yeah, you have a question? Yeah, it's just there's a hemisection. So it's not cut away entirely, it's just sort of, they removed a small sliver of the testes.
So that's where the sperm come from.
Yeah. So sperm come from the seminiferous tubules, which are here. We'll go into more detail on what happens in these seminiferous tubules. But we're looking at a cross-section of the tubule here. You can see it looks like a stratified cuboidal epithelium, and there's a lot of sperm cells that can divide, and then they emerge in the lumen here, where they mix with testicular fluid, and they kind of float around in that fluid. But eventually this testicular fluid mixes with the rest of seminal fluid to form semen. Now, these seminiferous tubules contain two main types of cells, you guys. Remember, the sustentacular cells and a population of dividing germ cells. Remember, the sustentacular cells are also called sustentocytes, and they help form that blood testis barrier. What was the function of this barrier? To protect the body from sperm, right? You don't want sperm wiggling anywhere else other than the system of tubules in the reproductive system, right? What's the other function of the blood testis barrier? Actually, you're right, these sustentacular cells do nourish the developing sperm, but they also form that blood testis barrier, right? Because remember, sperm are considered as foreign cells to the immune system, so that it also helps keep other immune cells out of the seminiferous tubules to prevent adverse immune reactions with those sperm cells. And by the way, the female body also considers the sperm cells as foreign, and you do get immune reactions to sperm cells, which is interesting. Now, a population of dividing germ cells you also find in the testes, and these actually produce sperm, and it begins at puberty. Now, nearby in the testes, you guys, not in the seminiferous tubules, but nearby, we have what are called interstitial cells. It makes sense because they're in the tissue. Not in the seminiferous tubules, but they're in the tissue nearby. These interstitial cells, also called the cells of Leydig, these produce androgens like testosterone. So that in response to hormones like FSH and LH, these interstitial cells can actually start to produce androgens like testosterone. And that's where testosterone comes from in males. So in terms of the process of spermiogenesis, or the formation of sperm, remember it occurs in the seminiferous tubules. Where do you find those seminiferous tubules? In the testes, right? Specifically in a place in the testes, the lobules. You got. So what separates the lobules? The septa, which are made of tunica albuginea. You got it. So what we're looking at here, you guys, is basically a cross-section through a seminiferous tubule. So you're looking at just a piece of the seminiferous tubule here. So this is the inside of the tubule right here. This is the rest of the testy tissue, which has your interstitial cells. What was the function of these interstitial cells again? They produce androgens like testosterone. You got it. And they do that in response to other hormones like FSH and LH. Now, over here we find some basement membrane, which is, like a connective tissue layer of fibers. And then attached to that we have our basically the wall of our seminiferous tubule. Remember, our seminiferous tubule has two main cell types, a sustentacular cell or sustentocyte, and a population of germ cells, which are dividing and give rise to sperm cells. So do you guys notice this large purple cell right here? that all of this purple cell is actually a sustentacyte, or a sustentacular cell. And what's really weird about these cells is they actually surround the dividing germ cells. So you guys notice here, right at the basal surface of the seminiferous tubule, we find something called spermatogonium, which is basically a germ cell that divides through mitosis. One of these daughter cells stays as the germline of cells, and it makes more sperm cells in the future. The other daughter cell we call a primary spermatocyte, and this primary spermatocyte then undergoes meiosis, which is 2 rounds of cell division, and from this primary spermatocyte, you get 4 spermatids. Each of these spermatids contains 23 chromosomes. So remember, we said that humans have 23 unique types of chromosomes. That's what these have, you guys. So each sperm cell here has 23 unique chromosomes. So you're going to have 1 through 22 autosomes, and then 1 of the sex chromosomes. Because it's male, one of these sperm will contain either an X, and the other one will contain either an X or a Y, right? So one of these sperm will carry an X, other sperm will carry a Y, including the other 22 autosomes. And depending on the type of sperm that fertilizes the egg, that determines the genotype of the zygote, right? So if you have a sperm that has 22 autosomes plus an X chromosome, and that is the one that fertilizes the egg, then that's what's going to make a genetic female, because now you have XX, right? Because the egg always carries an X. But what if you have a sperm that has 22 autosomes and a Y chromosome, and that's the one that fertilizes the egg? Then that creates a genetic male, exactly, because now you have an XY zygote. Yeah.
So would you create 50-50 on your XYs?
Yeah,
again?
absolutely.
That doesn't mean that 50-50 get released into.
Well, for this population of sperm that are developed, you will have 50% that have X chromosomes and 50% that have Y chromosomes. Now, the X chromosome is a little heavier than the Y chromosome, so these sperm actually weigh a little differently. And so there's interesting techniques where, let's say if you wanted to choose the gender of the sex of your child, you could actually put semen into a centrifuge where it spins down really fast. Like blood, the heavier elements will sink towards the bottom. Remember we had our red blood cells that were at the bottom? Well, the X-containing sperm would also sink towards the bottom of that test tube, and the Y-containing sperm would be more likely to be up top because they're lighter, because the Y chromosome is lighter, which means you could draw from a population of those cells to help determine the sex of your child. Now, that's more of the old school way to do it. Nowadays, what we can do is actually just harvest eggs from female, do in vitro fertilization, which basically means fertilization in a dish, like a petri dish. And then what you can do is once those eggs have been fertilized and zygotes start to form, you can sample cells from the zygotes to determine the sex of that child. And if you want, you can choose a particular zygote. That's what they call designer babies. And we'll get into this more so in pathophysiology. But it's kind of interesting, you guys. Now there are technologies where people can not only choose the sex of their child, they can also select for things like eye color and hair color.
What?
Yeah, so it's pretty, and we're just getting started. Like we can enter a pretty wild future where you might be, yeah, it's slippery slope, but it's interesting. Now the function of these sustentacular cells or sustentacytes is to form this blood-testes barrier, but they also nourish the dividing spermatocytes, right? So we have our primary spermatocyte that then divides through meiosis to form 4 spermatozoa here. And these spermatozoa then mature through spermiogenesis into what we talk about as being looking like sperm. Looks kind of more, like a tadpole where you have a head, a body, and a tail section. This all occurs within the seminiferous tubules and occurs on the order of millions per minute. That's how many sperm can be created. It's tremendous. Now, what we see here, then, you guys, is spermiogenesis, where we have our late spermatid that actually can divide into, or not divide, I'm sorry, just kind of reform into what we think about as a sperm cell looking like. But what's interesting, guys, is that the sperm cell doesn't have a lot of organelles. It's basically got a head section, a midpiece, and a long flagellum here. Okay, but they all come from one large cell here, which is a late spermatid. Now, what's interesting, you guys, is that what's left behind in this spermatozoan here is that in the headpiece you find a nucleus containing the chromosomes. You find an acrosome, which has digestive enzymes to break down the wall of the egg. We have a midpiece, which has a coiled mitochondria to make ATP. And the ATP that comes from this coiled mitochondria powers the flagellum. But that's it. In fact, what's interesting, guys, is that the sperm only brings in a nucleus to the egg. There are no other organelles that are brought in by the sperm. So what that means is that for the organelles that are in your body, especially mitochondria, those come from egg, not the sperm. So all the mitochondria in your body came from your mother's egg, not the sperm. Because when the sperm fertilizes the egg, it's only the headpiece that makes it through, and it just basically delivers in the rest of those 23 chromosomes to the egg to make the zygote. That's the male's contribution to the zygote. Otherwise, the mitochondria come from the egg itself, which is interesting. Now, in terms of stages of spermatogenesis, you guys, what we find is that our spermatogonium is basically the germline cell here, that we find 23 pairs of chromosomes, but it's diploid, which means we have 2 copies of each chromosome, right? So 46 total. Now, the male genotype is XY, right? Now, because these cells are dividing, at some point, one of those cells is going to get the Y chromosome, and the other cell is going to get the X, right? So that means that you have a population of sperm cells that have 22 autosomes plus a Y, and another population of sperm cells that have 22 autosomes plus an X. Usually, that's what should happen, right? And so then, depending on the type of sperm that fertilizes the egg, that will determine the sex of the child. So if it contains a Y chromosome, that will be male. If it contains an X chromosome, then it'll be female, right? But what if these chromosomes didn't separate out properly? What if you had a sperm that maybe had 2 X chromosomes instead of only 1? That means that when it fertilizes an egg, then you have an extra copy of an X. We call it trisomy X, and that causes pretty severe developmental problems. Or what if you had a sperm that was missing a chromosome, like missing an X chromosome, but it had 22 autosomes, and that was the sperm that fertilized the egg? Then you have a zygote that only has 1 X chromosome and no other sex chromosomes. That's called Turner syndrome, and Turner syndrome is just XO. We only have 1 X chromosome, and then we'll talk about this kind of stuff in pathophysiology. But that actually creates certain congenital anomalies, and you guys hear about like spontaneous abortions occurring. Sometimes spontaneous abortions occur because of these sorts of types of fertilization, where you have a mismatch of chromosomes and it doesn't create a viable fetus. In fact, most cases of Down syndrome, where you have an extra 21st chromosome, those actually don't make it to full term. Most cases of Down syndrome are spontaneously aborted at some point during development. But for the few that do make it, then we call it Down syndrome. They have an extra 21st chromosome, which you might wonder, hey, that sounds awesome, right? I got an extra chromosome. Well, the problem, though, is that because you have an extra chromosome, it actually causes gene dosing, where you have too much expression of certain genes, and it actually causes abnormal development. And so there's certain signs and symptoms that are consistent with that condition. We'll talk about that later. But so what this is showing you guys is just normal development of these cells. So normally the spermatogonium should divide into a primary spermatocyte. And this primary spermatocyte should undergo meiosis to make secondary spermatocytes that then divide into spermatids, which should have 23 chromosomes. That's a complete set, right? But it only has half of the number of copies. Normally you have 2 copies of each chromosome. So our sperm has the half there, 23. Egg has the other half. So when the sperm and the egg converge, then that creates a full set of chromosomes. That's why it's now a viable organism. Now, we call this haploid if it has half the number of copies of chromosomes. But it still has a full set of 23, just half the number of copies. So some of these will have X, some of these has Y, right? Now, after these spermatids have been formed, what happens is that these spermatids undergo a process known as spermiogenesis, where effectively they turn into spermatozoan or sperm cells, and they can leave the tubules and mature in the epididymis. Now, in terms of the male reproductive system, you guys, what happens is these sperm cells actually leave the seminiferous tubules through something called the rete testis. From the rete testis, they enter something called the efferent ductules, which enters the epididymis, remember? So the epididymis was the storage site for sperm. During an ejaculation, what happens then is that those mature sperm in the epididymis then leave through the ductus deferens, right? How do they exit the epididymis? What do you guys think? It's a muscular tube. You got it. It's actually smooth muscle contractions. You get strong waves of peristaltic muscular contractions that basically squeezes that fluid, testicular fluid, with sperm cells in it, through ductus deferens, up into the pelvic cavity, back behind the urinary bladder, where it then meets up with the seminal vesicle, right? And it enters the ejaculatory duct. And then from the ejaculatory duct, then it enters the urethra. And then we have 3 types of the urethra. We have the prostatic, membranous, and spongy urethras. And then this is the order of sperm movement through the system of tubules here. Now, just to summarize this process, you guys, what we find then is that we have our testi. Remember, the whitish tissue that surrounds the testi is tunica albuginea. It's separated into lobules here. and those lobules contain seminiferous tubules that then all connect at what's called the rete testis, which then connects to the epididymis through our afferent ductules. Now those go into the epididymis here, which is the storage site, and then from the epididymis we have the vas deferens, which then goes back behind the urinary bladder, meets up with the seminal vesicle to form our ejaculatory duct. From the ejaculatory duct, then it goes in the urethra and beyond. Right. So that's the order. That's the order of sperm flow. And you can see this sort of a summary of this process here.